Here we have the first standard that I will be teaching. It is that interphase consists of three phases: G1 phase, S phase, and G2 phase. The G1 phase is the most important because if it doesn't pass, it goes to the G not phase, which will consist of apoptosis, and the cell will die. So G1 phase consists of proteins being made so that the cell can grow and be ready to replicate the DNA. And then S phase is when the actual DNA replication happens and it synthesizes new DNA. And then G2 phase are where the checks are made so that ensures that the chromosomes are ready to be split through mitosis. So there are three checkpoints in the cell cycle. The first one is the cell growth checkpoint, which ensures that the cell has the right proteins and is able to synthesize later on. So this is the most important because if it doesn't pass, it goes through apoptosis. The second one is during the S phase, and it's the DNA synthesis checkpoint to make sure that all the DNA has been replicated correctly. And the third one is a mitosis checkpoint, and it ensures that the cells are ready to divide and the cycle repeats. Here we have a little visual on the cancerous cell and a normal cell. So essentially, a cancerous cell will keep on dividing even though it has damaged DNA. The damaged DNA is called an oncogene, and it causes uncontrolled cell growth. And there's three ways that this can happen. One is movement of the DNA within the genome, which is like a new promoter. Amplification of the proto-oncogene, which has three copies of the gene sequence to produce an ample amount of proteins so it don't keep on dividing that way. Or the point mutations in a control element or proto-oncogene itself. So essentially that could be like not having a stop codon. So then it would keep on making the protein. Here we have a diagram for the cyclins and the CDKs. The cyclins are proteins that are made to activate the CDKs, which will then phosphorylate the targets and cause the certain phase that it's in to occur. So essentially here we have the G1/S cyclin, which will attach to the CDK, which will phosphorylate the targets and cause them to be activated, which will then have S phase begin as a result. And here we have the M cyclin, and the M cyclin has a low amount throughout the whole thing until it gets near to the M phase, or mitosis, if you will. And this will result in a spike in the M cyclin so that the mitosis can be activated. So M cyclin attached to CDK, targets are phosphorylated, and then M phase begins with the spindle forms, and then the chromosomes condense, and the nuclear membrane breaks down so that the chromosomes can be split equally. Here we have a diagram for mitosis. We can see the centrosomes with the spindle fibers and the chromosomes inside the nuclear envelope. Step 1. Step 2, the centrosomes go to different sides of the cell, and they use their spindle fibers to align the chromosomes like this in a straight line so that they're equally split. So here the spindle fibers pull apart and they split it up, and this is when cytokinesis is going to occur, and the new nuclear envelope envelops the chromosomes, and two identical daughter cells are created. Mitosis relates to asexual reproduction because it occurs in somatic cells, in the body cells, and it has a genetic information of only one person.